Hello everybody, welcome back to MMBS-111 Introductory Microbiology. This mini lecture is going to cover content from section 3.3, particularly about the cell envelope. There are 3 learning outcomes that you should be aware of as you work through this mini lecture. First, be able to explain the major cell wall differences between gram-positive and gram-negative bacteria. Name 2 functions of the cytoplasmic membrane, and describe the component of the gram-negative cell wall that provides protection against certain antimicrobials. So let's talk about the cell envelope. The cell envelope is the outer layer of the cell and is typically composed of either 2 or 3 layers depending on the species. The cell wall, the cell membrane or cytoplasmic membrane, and then in some bacteria we also have an outer membrane. The cell wall is the structure that helps determine the shape of the bacterium. It is very strong and rigid. It helps to provide support to the bacterium. It prevents it from bursting or collapsing under differing osmotic pressures within the environment. Certain antibiotics and other drugs are used to target particular components of the cell wall, and that disruption then helps to sort of disrupt the integrity of the cell wall, causing the cell to lyse or to rupture. There are some significant differences between gram-positive and gram-negative cell walls, as you can see here in the image. On the left, we have the gram-positive cell wall, and you can see that we have the cell membrane here, and then we have this very thick layer of peptidoglycan that composes the gram-positive cell wall. And then we have lipoteichoic acid molecules that sort of integrate in between the peptidoglycan layer. In gram-negative cell walls, we also have the cell membrane, but then we have a very small, either single or double layer of peptidoglycan, and then outside of that we have this outer membrane. And this outer membrane is composed of lipopolysaccharides that act as receptors. They also produce an endotoxin, which contributes to the symptoms of gram-negative infections. It can cause fever and shock. Some other components of this outer membrane are the protein porins. These are protein channels that allow very specific small molecules to enter the outer membrane. So these are the primary differences between these 2 cell wall types, and their differences contribute to their staining. So the way that we distinguish whether an organism is gram-negative or gram-positive is by staining a smear. And we begin by staining first with crystal violet, and the crystal violet stains both cells this purple color, and the stain adheres to the peptidoglycan layer of both cell walls. Then we add Gram's iodine. Gram's iodine is a mordant. It helps to fix the stain to the cells. And then we add alcohol, and the application of the alcohol serves to decolorize any gram-negative cells. So the alcohol dissolves the lipids that are components of the outer membrane, and this allows the dye from the peptidoglycan layer to be removed. So after the discoloration step, then we add a second dye, safranin, which is a red dye, and our gram-positive cells remain purple. The dye fixes to the peptidoglycan, but that violet-purple color overshadows the red. However, the gram-negative cells are stained red. Since the color was removed, that colorless cell is going to accept that red stain. We also have some other cell wall types. These are not common, they're atypical, and one type is the acid-fast. And some examples are Mycobacterium and Nocardia. Acid-fast cell walls have the cell membrane and then this layer of peptidoglycan, and then outside of that they have a layer of long-chain fatty acids called mycolic acids. And these are only found in the cell walls of acid-fast bacteria. And mycolic acids increase the pathogenicity of these organisms because they are resistant to certain chemicals and certain dyes that might impact other cell walls. This is an acid-fast staining procedure, and we begin by staining the smear with carbylfusion, and we get this bright pink color. And while the stain is on the slide, then we heat the slide, and this helps to adhere the stain to the cells. Then we have a decolorizing step where we wash with an acid alcohol, and any organisms that are not acid-fast will lose their stain. And then we restain with a methylene blue. So non-acid-fast organisms will be a blue color, while the acid-fast organisms will be a pink color. Archaea also possess atypical cell walls, but there's a large variety of cell walls found in the kingdom Archaea. So we have some that are composed completely of polysaccharides and others that are composed completely of protein. All of the Archaea cell walls lack true peptidoglycan structure, so none of them possess peptidoglycan within their cell walls. And some Archaea are cell wall deficient, meaning they completely lack a cell wall. Mycoplasmas are a group of bacteria that are naturally cell wall deficient. They do not have a cell wall. Their cell membrane is composed of sterols that help to stabilize the organism so that it resists lysis, unlike other cell wall deficient bacteria that might be more prone to rupture. They are very small, about 0.1 to 0.5 micrometers, so they're one of the smallest bacterial types. We also have L forms, and these are variants of bacterial species which over time have lost their cell wall. And this loss can be stimulated by treatment with a cell wall targeting antibiotic like penicillin. These L forms, scientists believe, may play a key role in persistent infections that continue and seem to resist treatment. These L forms lack a distinguishable cell shape, so we can't say that they are bacillus or cocci. They're generally described as spheroid. Let's take a look at the gram-negative outer membrane. So this membrane is similar in construction to the cell membrane. However, there are a couple of components that are new, and the first is those lipopolysaccharides, and these act as receptor molecules. As I said before, they also produce an endotoxin. That this endotoxin increases the virulence of these gram-negative organisms. And they also contain these porin proteins that allow certain chemicals to penetrate that outer membrane and then enter the cell. There are a few differences in cell envelope structure depending on the type of organism. So the outer membrane of the gram-negative bacteria acts as an extra barrier, and it is particularly impervious to antimicrobial chemicals. So gram-negative organisms tend to be more difficult to kill than gram-positive bacteria. The use of alcohol has been very effective in addressing gram-negative organisms outside the body. So this is why you use an alcohol swab to cleanse the skin before certain medical procedures. Also, particular antibiotics are used in the treatment of gram-negative bacteria that attack that outer membrane. So this is the structure of the cell membrane, or the cytoplasmic membrane. This structure is consistent across all groups. So archaea, bacteria, and eukarya tend to have very similar structure of the cell membrane. It's composed of a lipid bilayer. As you can see here, we have our hydrophilic heads and our hydrophobic tails on the inside. We also have a number of carbohydrates and proteins that tend to float within this matrix. The cell membrane is the site for a number of different activities, including energy reactions, the processing of nutrients, and the synthesis of different components and compounds. The cell membrane is also key to regulating the transport of nutrients into the cell and waste out of the cell. The cell membrane is called selectively permeable because it is very selective about the types of materials that are allowed to cross the membrane and either enter or leave the cell. And that is the end of mini lecture 3.3. Make sure you go through the questions here and be sure that you can answer them correctly. Remember, you can go back and review this multiple times, and hopefully it was helpful.